fantastic lineup of speakers. So exactly, I was about to say that this meeting will be recorded for posterity. So if you don't want your image to be recorded alongside the slides, please turn off your camera. And as well, please mute your microphone while the speakers are presenting, and just turn your microphone on before asking a question, basically. So welcome. This is like the fourth workshop organized by Easy Rider, and I'm happy to be leading it. I am Luca Pegoraro. I'm a postdoc at WSL in Switzerland, working in automated pollinators monitoring. And before we start, I want to give a quick shout out to the WSL Biodiversity Center that supports biodiversity activities related to all areas of research, and it's been instrumental in building the network that this workshop is based on, and the UK Centre for Ecology and Hydrology, and especially Alba Gomez-Segura, that is leading the workshop together with me, and Tom August, and of course as well the Wide Labs platform that is kind enough to host us and provide an excellent platform. More on our group later. So here is a rundown of the program. We have 3 talks with a very different take on this problem from different speakers. And after each talk, there will be a chance for you to ask questions. You can either raise your virtual hand and we will give you like the word and you can ask a question directly, or you can type it in the chat and we will read it. After the 3 talks, there will be a discussion in which we will separate in breakout rooms, and a message will pop up on your screen and you just have to click on it, and there will be somebody in the room that will basically guide you through the discussion around the 4 main topics that we have selected. After that, we reconvene and we discuss our findings in the different groups that we have, and have a discussion of where do we want this technology to go and so forth. The whole thing should take more or less 2 hours. So without further ado, I will move on to our first speaker, which is Claire Carvell. She is a senior ecologist at the UK Centre for Ecology and Hydrology. And Claire, I'll leave the floor to you.
Thank you, Luca. I'm just, there we go. Well, good afternoon, everybody. And yeah, I'll start by thanking Luca and Tom and Alba for the invitation to speak this afternoon. It's really exciting to be part of this Easy Rider workshop. I'm Claire Carvell. As Luca said, I'm an ecologist at the UK Centre for Ecology and Hydrology. I have been conducting applied research there for just over 20 years. A lot of that spending time looking at responses of bees, bumblebees especially, to landscape change. But since 2015, I have been coordinating the design and the setup and the implementation of the UK Pollinator Monitoring Scheme. So I'm really pleased to be talking about that today and just touching on some of the opportunities and challenges for integrating automated methods. On to my next slide. Okay. So yeah, I'll start by just talking more generally about the wider challenges of designing a national systematic monitoring scheme for pollinating insects, particularly touching on what are we trying to measure, these questions that are really critical to designing a scheme that's fit for purpose. How are we going to measure it, and who, what actors should or could be involved in the process of collecting that data. I'm then going to talk to you a little bit about the UK and EU pollinator monitoring schemes, and then touch on these opportunities and challenges for automated monitoring, which I've kind of divided broadly into scientific, practical and moral opportunities and challenges. And I hope the overall message will be that there's massive potential for the integration of automated methods, but for these to augment and complement rather than to replace traditional monitoring schemes. I'm going to be focusing, of course, on flower visiting bees, bumblebees and solitary bees and honeybees and hoverflies. But the methods that I'm talking about are involving capturing the whole flower visiting insect community. And that, of course, includes butterflies and moths. But the main focus will be on the Hymenoptera and the Diptera. But just to go back onto the butterflies and moths and reflect on some of, I think, what is globally probably the most powerful data set, a long-term monitoring data set for biodiversity, perhaps anywhere in the world, which is Britain's larger moths data set from the moth trapping survey and from recording of moths across Great Britain. And even there, where we have a really large distribution of volunteer recorders and trapping efforts as part of this scheme, we still see a lot of fluctuation and variability in patterns of both abundance that you can see on the left over time and in patterns of distribution. So even now, with one of these most powerful data sets globally, it's still showing us results that need careful interpretation, that may sometimes be inconsistent and make it quite challenging to describe these complex drivers of change. So with that in mind, what do we have for bees and hoverflies? Well, in the UK, most of the trends that we understand to date have come from long-term opportunistic records. So these are records of the presence or absence of species that are sent in to our recording schemes, our national recording schemes by amateur expert volunteers. But of course, because they're recording where they choose and in areas they choose and when they choose, these data tend to be very noisy and they're effectively unstructured. But we have very highly developed models or occupancy modeling approaches by which we can start to account for some of that variation and bias in recording effort. So some of you may be familiar with the trends that have been plotted from these occupancy data sets. This is a sort of combined index of occupancy of Great Britain of all the pollinating species for which there was enough data. So about 377 out of our total of sort of 550 odd bees and hoverflies in the UK. And so we can look at which of those species are increasing or decreasing in their distribution across Great Britain. And indeed, we've been developing recently some exciting new tools, for example, the Robit tool, to allow us to actually understand better what the risks of bias are in some of these studies of temporal trends using occupancy data. But indeed, another recent paper where we've looked a bit further afield, this is some data from the neotropics in South America. In fact, inferring trends is not always improved simply by adding more data to your models. So in this particular example, we looked at an amazing 6-fold increase in the number of bee species that have been recorded on GBIF for Chile. But we found that, again, using the Robit tool, most of the biases, spatial biases, geographical biases, temporal biases in those data remained, or in fact, in some cases increased, because of the nature of the recording remains the same. And so actually, when we looked at relative indices of change in these Chilean bees over time, we find little difference in the result that we get from a sort of data poor versus a data rich scenario. And that takes us on to effectively the need for more systematic or standardized monitoring, which again, I'm sure is no kind of novel concept to most of you. And I just wanted to say something about the themes and the concepts that we had in mind when we were designing this new systematic monitoring scheme for pollinators in the UK, and which have now been applied to the EU situation as well. So we wanted to start, and I think it's really clear to think firstly about the outputs or the metrics. What measures do we actually want to be dealing with here? What do we want to find out? So they may be connected with pollinator population status or health, or the pollination service itself, or measures of the capacity of the landscape to actually support pollinators, which we might use as a corollary of pollinator health itself. And it's important we can think about all the different elements of the framework that are needed to deliver those outputs or measures. So we thought about very much about the recorders who might be needed to collect the data, the methods they were going to employ, the sampling site networks that would be required to have the power to deliver the outputs we wanted. And then what support tools do we need in order to make all of this happen? And again, you can already see how this is a framework that's put together for a traditional monitoring scenario. But of course, most of these concepts are going to be relevant in the sense of automated monitoring as well. So in the UK case, we spent around a year doing quite a wide consultation on the suitability or the feasibility of different methods and different people, who's doing what and who do we need at what point in the chain to, again, provide the data on these different metrics and measures of interest. And we also had a campaign in the field of testing and trialing the different methods. And we ended up in 2016, 2017 with what is now the UK Pollinator Monitoring Scheme. So the scheme is, I think, still probably the world's first monitoring scheme that's been running since 2017, that's generating systematic data on the abundance of bees and hoverflies and other flower visiting insects, and doing this at a national scale across the UK. Currently, POMS is running using 2 large-scale surveys. They're primarily run by volunteers, but it's an important mix of volunteers and professional surveyors to be able to measure changes across the UK in the way that we need to. So I'll talk briefly about the 2 surveys. We've got an observational 10-minute flower insect timed count, otherwise known as the FIT counts. And then there's a more intensive systematic survey across a series of 1 kilometre squares across the UK, which uses pan traps and records information about habitats as well. And very much key to the development of POMS is that it's underpinning the evidence base for our different national pollinator strategies in England, Wales, Scotland, and in Northern Ireland. And also critically, the scheme is set up to provide access to the data at full resolution and to eventually provide metrics and hopefully new indicators that will take us towards some of our environmental targets, such as our 25-year environment plan and the species abundance targets that DEFRA and so on are looking to develop. So the flower insect timed counts, this is a 10-minute count to collect data on the abundance of flower visitors and of course the plant pollinator interactions that are taking place across a variety of habitats and places. So you can do a FIT count pretty much anywhere in warm, dry weather from April to September. And the count involves counting all the insects that land on a patch of target flowers. And those target flowers are specifically from 1 plant species within a plot of 50 by 50 centimetres and recording what you see during the 10-minute period. And importantly, this is very much a citizen science survey because the insects that are being counted are identified just at group level. So, for example, distinguishing between bumblebees, solitary bees, honeybees, and then hoverflies, other flies, butterflies, moths, beetles, and so on. And excitingly, you'll see in the bottom right, we launched an app to go with the FIT count. So the FIT count app has been running since last year, 2021, and we've really seen an increase in the amount of data coming in as a result of having the app. So see here since 2017, an increase in the number of counts coming in. We've now got more than 8,200 counts over the 5 years of the POMS scheme. Most of these, vast majority, are from well over 1,000, nearly 1,500 volunteer recorders. And as you might expect, a lot of them are being undertaken in gardens, but also a large proportion out in the wider countryside and in various different types of habitat. So I'll talk now about the other survey that POMS runs, and this is our 1 kilometre square surveys. So these are more intensive surveys over a network of up to 95 sites that include Northern Ireland as well, as of this last year. And it's designed to detect broad changes across the UK, broad UK level changes, in other words, not necessarily country level change, but change in the abundance of insect groups and in some species richness of bees and hoverflies, species distribution through integrating with occupancy models that I'll mention later, and through recording flower abundance and habitats as well. And there's stratification, so around half the squares are in agricultural areas and around half are in semi-natural areas. So we've got a mix of different habitat types effectively across the UK. So this is a map of the squares that are currently covered by the POMS scheme, and I'll say something in a minute about why we have 75 to 95 squares. And importantly, the ones that you can see in blue are adopted by volunteer recorders. So we have fantastically committed volunteer surveyors going out 4 times a year, setting out pan traps and monitoring the insects on those sites that are marked in blue. And then we have a paid team of surveyors that effectively fill in the gaps and also provide training and mentoring to the volunteer network. And we're using some really nice descriptions of some of these fantastic places to try and encourage more people to come and take part and get involved. I'm just going to move on a little bit more quickly. I've spotted the time. So at the same time as the UK POMS has been developing, there's been a huge amount of work at the EU level funded by the Commission. And in particular, a group of experts led by Simon Potts put forward a proposal for an EU level pollinator monitoring scheme to operate across member states. And that was published in 2021. And I'm just going to quickly give you a flavour of what that's involving. So don't worry too much about the technicalities of the diagram. But again, if you look on the left, you can see that it's taken this focus again of the taxa. What do we want to record? What methods is the scheme going to use? What types of recorders are required? And what measures are going to result from that data and from that recording and monitoring? So there is use of standardised transects and standardised pan traps to sample bees and hoverflies in particular, very similar to POMS. And then the EU POMS is also covering these complementary approaches, for example, for rare and threatened species, for moths using light traps, and also looking at additional modules, in particular for pollination services, flower visitors, so it's expanding to the fit counts as well, and also understanding how Malaise traps might tell us more about wider insect biodiversity at a subset of these sites. And the project that's overseeing the trialling and testing of this approach, particularly the two blue bars on the left that you see referred to as the minimum viable scheme, this is referred to as the SPRING project, and that's also underpinned by a group of experts under the STING project that's coordinated by Simon Potts to provide additional interpretation and analysis of the data that's resulting. So how do these systematic or national level long-term monitoring schemes offer opportunities and challenges for integrating automated methods? Firstly, in terms of the sort of scientific perspective, I want to think about, again, what do we want to measure? And again, it'll be no surprise to you all that we're thinking here about species and how the different approaches to monitoring have different potential to tell us more about more or less other different types of species in the community. This is just some data that we took from the pilot phase of the UK pollinator monitoring scheme published by Rory O'Connor in 2019. And this is just to show, again, that it's important to understand the relative potential of the different methods that we're using to generate records across the species and across the different groups. So this is just showing us in terms of species richness that actually pan traps and transect walks were relatively on a par at equivalent sites in terms of the accumulation of species over volume. But what was really interesting was when we introduced a sort of researcher versus expert comparison. So researchers are sort of less highly trained, less expert entomologists versus long-term recorders and experts in the field of bee and hoverfly identification. And as you might expect, we found that in particular for the solitary bees in the middle and the hoverflies on the right, the experts undertaking observational transects performed just as well as pan traps that were set out on these sites, with the researchers undertaking observational transects performing slightly less well. And this led me to thinking of the question, so potentially could automated methods have the potential to bring us a lot higher up this curve? At what point is a given systematic monitoring scheme going to give us a sort of saturation level of species in a given community or at a given site? And I think this is something that's going to be quite a challenge to find out. In relation to the species, so what kind of species is POMS, UK POMS, currently sampling using pan traps? So we're tending to get move on to sample around 42% of the total list of UK bee and hoverfly species, and that's happened over the last 5 years of sampling across 75 sites in the UK. So it's a relatively complete list, considering that we know pan traps don't sample everything, but of course for many of those species we're only sampling them in fairly low numbers. There's a great opportunity here for automated methods to be complementing this approach, to potentially sample many more species, and potentially, of course, including night active flower visitors, because again, the pan traps are only being placed out during the day. But there's a question here, what taxonomic resolution, and I'll come back to that in a moment. With POMs, we also had to think carefully about the sample sizes and site networks in relation to the statistical power that we would have to detect changes, and also the cost of running the scheme. So again, just briefly, I don't want to dwell on the technical detail, but we looked at scenarios that would give us the statistical power to look at between 30 to 50% change over a 10-year period. And effectively, we're at this point in the table at the moment with a network of 75 sites. But indeed, with increased number of sites, again, potentially through remote methods of monitoring, potentially through automated methods, we could be increasing the coverage of the number of sites and then increasing our power to be able to detect changes either in more species or to detect changes in the species that we're already sampling at a greater level of power. So I'm just going to move on. I wanted to say something about understanding the effects of context, of whereabouts in the local or landscape level context you actually are sampling in these monitoring schemes. And this is a schematic that we've used in the Spring project, put together by Simon Potts, just thinking carefully about how the position of a trap that might be a pan trap or an automated monitoring trap actually affects the pollinators that it's designed to catch. So you've got traps that are effective to the pollinators. You've also got local patches of flowers that are also attractive to the pollinators. And of course, this varies with flower number in the local area and with flowers in the landscape. You then have the insects that are interacting between the flowers in the landscape and the pan traps or the sampling stations themselves. And these interactions can get quite complex. And of course, the pollinators we're looking at are changing and differing in their community composition, both in space and in time. And the flower types are changing as well. So critically, we want to be moving to a situation where we may have a long-term surveillance scheme, but how can we monitor interventions in the landscape? For example, agri-environment schemes where patches of habitat are being manipulated at a landscape level. And of course, other environmental factors come into play, like the time of day and the weather when we're trying to interpret all of this data. So I think these are challenges for both automated and traditional methods to better understand the effects of the context in which they're being placed on the patterns of change that we see over time. But there's maybe also an opportunity here for automated methods to particularly help with some more targeted monitoring of interventions. And that could be in urban or rural settings. And this could be happening alongside these long-term structured surveillance schemes. I want to say something briefly about data standards and the metrics again and the modelling frameworks that we're using, which I think are really important. It's probably a huge topic on the Easy Rider project is the volume of data. But we've now got some really exciting methods of modelling which allow integrated modelling. So not just looking at occupancy or occurrence records that I showed in the beginning on their own, but actually combining models, combining data layers from occurrence records and structured monitoring schemes. For example, on the left, you could see the bee wars bee data for the UK combined with the more structured bee walk data or POMS pan trap data for bumblebees, for example. And I think knowing how these integrated models have the potential to increase our precision, for example, for estimating trends, there's an opportunity there to augment the power of those models with additional data. Just touching on the practical challenges and opportunities that are at play. And again, these are probably obvious to many of you, but it's certainly played out with the UK POMS scheme. These are some of our fabulous sites, particularly in some of the more hilly areas. You can see in Wales there, the picture in the middle with the vehicle in the bottom of the valley, and then you've got livestock to deal with, cattle and sheep, potentially damaging traps, rivers to cross and forests to traverse. And again, you can see some quite mountainous areas there with Richard in the foreground. And these often are fairly remote and risky locations, which are really under-recorded. This POMS scheme is putting a lot of dots on the map in places where insects aren't currently being recorded through other methods or other schemes. And there's obviously the risk of damage either to the pan traps themselves or indeed to our surveyors. And of course, safety needs to come first. So there is potentially a limitation in being able to get volunteers to many of these sites. As it happens, in the POMS scheme, we've had thousands of trap stations now being set over the last 5 years, more than 4,000. And out of those, only about 2.5% have actually been recorded as being disturbed or damaged. So there is something to be said for having these relatively cheap, relatively unattractive systems which are unlikely to get tampered with. And again, that question about the logistics of putting a really expensive, amazing looking camera trap into a location where it may be vulnerable to damage. On the flip side, if we can get some of these traps into potentially hard to reach areas and have them continuously monitoring and being remotely operated, I would see that as potentially a really good opportunity. I can see the time's ticking on, Luca, so I wanted to just touch on this question about capacity to identify large volumes of samples, but I'm not going to go into a lot of detail here, apart from to say that I think we need to continue to recognise the expertise within our taxonomists and build on that, because of course they've got years of observational skill and ecological knowledge. And this could be embraced, I think, with the community of entomologists that are building up now and starting to come through, many more degrees available to support them and to help with validating the results. And the validation process of the UK POM scheme is a really important part of what we're doing to try and get really good quality data. I just want to finish on this question about the sort of moral or human opportunities and challenges around these monitoring schemes for pollinators. I think it's become very clear recently that there are huge benefits of engaging with nature, particularly during the pandemic recently. So a survey done by Natural England revealed that 84% of people found that being in nature made them happy, and large numbers had engaged more with everyday nature when they had a chance just to slow down and appreciate what was happening on their doorstep. And some work by my colleagues, Michael Pocock at UKCEH on the Nature Up Close and Personal project, which was also run during the years of the COVID pandemic, has shown really that the quality of citizen science and engaging citizen science is really good for you. And you can find out more about that, hopefully, on their website. This is a lovely photo, which actually I haven't animated it. I think I've put a box over the loveliest bit of the photo, but just to show POMS has attracted not only individuals, people wanting to do more in the outdoors, but families as well. And we've had some really wonderful feedback from people engaging with the monitoring scheme. I'll just leave you to read those. And I think my favourite one being someone, one of our volunteers saying that their POMS days were some of their favourite days of the year. And just going back to the flower insect timed counts, not only are we running the Fit Count survey and the Fit Count app in the UK, but we've now taken this out to, I think it's up to 9 or 10 other countries now are developing and adopting the Fit Count app. So not only across some of the European countries that are part of the Spring Project, but also in South America. And we're really excited here to be working with our colleagues in Brazil. They've even got Fit Count bags, Fit Count t-shirts, Fit Count quadrats, Fit Count pencils, and have really embraced this as a means of forming this global network potentially of collecting standardized data on insects and flowers. And of course, this data does rely on citizen scientists, again, which is another whole discussion. And the automated methods have definitely got a place here, I think, to augment, and there's an exciting role to play in interacting with that citizen science data. But I think if we need to really make sure that this opportunity for connecting with nature is retained, again, through keeping people involved. Otherwise, we kind of effectively risk losing our connection with the very things that we're actually trying to save. Before I finish, and I'm sorry I've overrun a little bit, but I just wanted to highlight this fabulous paper, which I think summarises a lot of the points I've actually tried to discuss a lot more eloquently. I only just discovered this paper this week, but this has just come out. And so I'd really encourage you to have a read of that tree review by Van Klink and Tom and other colleagues. Thank you, as always, to the POMS team, our funders, landowners, the volunteers who collect the data, my POMS colleagues, CEH colleagues on the left. And for anyone who's keen to find out more, the POMS email address is down there, and the website address. And also anyone who wants to find out more about the EU work under the Spring Project, you can contact David Roy or Simon Potts. Thank you.
Thank you, Claire, for a fantastic talk. I think we don't worry too much about the time and we eat away a bit to the discussion time. That's completely fine by me. So we can take a couple of questions from the audience. Claire, there is already one in the chat from Hien. He asks, how can we from Vietnam participate in the project? Can you send us some drops for sharing the design so we can build it here? Perhaps you can answer more generally to participating in the project internationally.
Yeah, so that's a great question. Obviously, the UK POMS project is funded at a national level, and we're just moving into a phase of being coordinated through JNCC, and the same really with the EU project. As a scheme, the funding mechanism and the running of that is happening at that sort of national or EU continental level. But we'd be happy to talk about if this colleague is maybe referring to the Fit Count app. Happy to have a conversation about extending the Fit Count app to any other countries outside of Europe and South America, because we've definitely built the infrastructure within the app to allow us to do that. So yeah, please get in touch with the POMS email address.
Thank you. And if there is, yeah, oh, okay, they are coming in now. So Jamie asks, completely agree that automated methods need to complement existing methods. Don't want to shift that baseline any further. So actually, that's exactly what I wanted to ask. Well, did you consider this problem of the shifting baseline in your analysis of the power you needed to detect a certain percentage of change over time?
I think the answer to that question is probably no, but I don't want to exceed my technical ability in answering a question about power analysis. So I think the problem with undertaking a power analysis 6 years ago was, or 7 years ago now, was that we actually had very limited real data from which to understand levels of variation and to properly parameterize those power models. Again, the power to detect change with 75 sites is very much indicative. And we're excited to maybe take a position perhaps next year where we'll have 6 years of data to actually rerun that power analysis and understand whether it's accurate and whether we're running true to our predictions about being able to detect change.
Mm-hmm. Great. Any more questions from the audience? Maybe we take another quick one if there is. If not, please feel free to type the question in the chat and Claire will try to answer it in the chat or during the discussion afterwards. So I think now we can move on to our second speaker, Maximilian Sittinger, PhD student at the Julius Kuhn Institute, and is going to present to us DIY camera trap for automated monitoring of flower visiting insects. So thank you, Max, and take it away.
Okay, thanks, Luca. I hope you can hear me now.
Yep.
So, thanks for the introduction. Yeah, we are working in the Monvir project, which has a goal to develop new methods for insect monitoring for a longer time period in Germany. And during this project, we developed this camera trap. We call it do-it-yourself camera trap because it is relatively easy to build and program, and we think it's possible also for the community to pick up our design and maybe use it also for pollinator monitoring. So we are building on a lot of work that was previously done in different working groups. Especially the work of Kim Bjerge and the Tokohoye group really inspired me to develop a similar system, which is more adapted to flower visiting insects compared to night active insects. So for the core hardware, we have three components. The first one is the Luxonis OC1 or OpenCV AI kit. This is basically a 12 megapixel camera sensor that is directly connected to an Intel Movidius Myriad X VPU chip or visual processing unit. This means that you can do on-device CNN inference, so you can run.
Max, I don't think your slides are flipping.
Oh.
No, we are seeing just a normal PowerPoint. Yeah.
Yes.
Is it working
now?
now?
Yeah,
Yep.
now it's
Now
working.
it's
Yeah.
perfect. Sorry.
So the also the hardware slide, the correct slide. Okay. So you can run object detection, but also classification models directly on this OR camera with a relatively low power consumption. The camera is connected to the Raspberry Pi CO2W, which acts as host for the OC1. This means that the software pipeline in form of a Python script gets sent to the camera, and then the Raspberry Pi only receives the processed data. So for example, the images. So there's no load at all on Raspberry Pi, which makes it possible to use the 0 version with a limited resource capacity. The Raspberry Pi is connected to the Pi2 head via the GPU pins. And this board can be used for power management. It has an onboard real-time clock. And most of the time it stays in a low power deep sleep state, but you can program a wake up alarm. So the Raspberry Pi and the whole system gets wake up at specific times and runs, for example, the detection pipeline and then shuts down again. And this makes it possible to use it for continuous monitoring without needing to go to the camera trap and configure anything. So the whole setup is powered by a 9-watt solar panel, which is connected to a 12,000 milliampere hours battery. And this battery charges a second battery, the official Pi2 battery, but you could also use only the Pi2 battery. So these two batteries makes it possible to use it for longer times without sunshine, but one battery is totally fine. So the power from the solar panel charges the batteries. This is used for the whole system to work, and the all cameras is fit in the lid and connected to the Raspberry Pi. All of the electronics are fixed inside a waterproof enclosure, and a silica gel pack makes sure that there's no moisture buildup, for example, because of temperature differences between night and day. And we also added a mounting fixture that you can mount it on, for example, a standard tubular post, which is also used for science or something like this. The cost for the minimal setup, this means there's only the Pi2 battery, a smaller SD card, and no mounting options, is about 500 euros. For the full setup that you can see in this picture, it's about 700 euros. So it's relatively cheap compared to other trap designs, and maybe this makes it possible to use or to deploy more than one trap for several field sites. For the image annotation, to train our detection models, we use RoboFlow. This is an online platform that is free to use. For our base model that we are using at the moment, we only annotated 446 images with a total of 436 insects. So maybe if you're familiar with these data sets, this is not much. So the model we are running at the moment is very basic. And I think it would be good if we get more training data for better models, basically. So, but we are using only one class. So for the detection, we are detecting all insects. We don't separate them into any taxonomic classes at this step. We chose the smallest YOLO v5 architecture, the YOLO v5 nano models. And from the training of on this data set, we got an MAP of 0.86, which is maybe not the best, but at the moment good enough, at an image size of 416 pixels. So yeah, the model training was done with the Google Colab notebook. So Google Colab is a platform where you can use a free cloud GPU or graphics card for fast training. And we will also publish this notebook together with all of the Python scripts and documentation. And in my opinion, it is really easy, if you don't have any prior experience, if you use maybe open source data set to train your own model, you can do this with these notebooks directly in your browser without needing any special hardware. Also in the same notebook, we have to do 3 conversion steps. So first, we'll export the model to the ONNX format. From the ONNX format, we can convert it into the OpenVINO format, which is also optimized for smaller resource-limited hardware, like, for example, the Intermo videos. And from this OpenVINO format, we can convert it to a blob file. This is only a 3.5 megabytes file, which basically has the detection model and can be directly used by the OCamera in the Python API. So for the model deployment, we just have to set the path to the model and to the config JSON that gets automatically created together with the model. And this makes it really easy to switch different models or to try out different models because you only have to change these two lines in a Python script, and all of the rest gets automatically set by the program. We are also combining the detection with the object tracker from the OpenVINO toolkit. This is necessary because otherwise on each frame, every detection would be counted as new insect. And with this object tracker, you can set a tracking ID. This will follow also a moving insect. And with this, you can differentiate the individual insects in each frame. The object tracker is based on the Kalman filter together with the Hungarian algorithm. I think this is the for most object trackers, these are the algorithms that I use. And it uses short-term tracking, which extrapolates the object trajectories from previous frames. So this means even if there is a low neural network frame rate from the detection model, the object tracker can track between the frames that are not detected by the object, by the detection model. So with our current model, together with the object tracking, we reach about 4 to 5 frames per second, which is enough to track most insects. There are some, for example, some wasps that are flying really fast. There are still some problems, but most normally moving insects will be correctly tracked. In this Python script, you can also set the camera output. So we are using a 4K sensor resolution, but this would be way too high or too high of a resolution to use as input for the detection model. So we are downscaling the full field of view of this high resolution frame to 416 pixel and using this as model input. And in parallel, we are running a high quality stream, so the full 4K stream, which gets synced with the model output or the detections. This slide, I try to visualize it a little bit simpler. So we are starting with the 4K camera resolution, downscaling this on the OAK device to 416 pixels, which is used as input for the detection model. The detections are used as input for the object tracker node. And these, so the pass through detection and the tracklets from the object tracker get synced with the high quality frames in a script node, also on the device. And synced frames or the crop detection from the high quality frames together with the tracking IDs and information on the detections are sent to Raspberry Pi Zero, which only has to save them to SD card. So there's no load on the Raspberry Pi except from saving the processed image data. On the right side, you can see the model inputs. So these are the low quality frames that are squashed to square, so to 416 pixels. And in the middle, you can see the high quality frame, so the 4K frame, and the bounding box coordinates from the low quality frames basically gets synchronized with the high quality frames. And from this high quality frame, you can crop the detections, save them, and these can be later used for classification. So this is the most important output of this pipeline. Together with the crop detections, we are also saving for each recording interval all of the metadata that we get from the object tracker and the detection model to a CSV file. So the timestamp for each detection, the label together with the confidence. So you could also use a model with more than one class, that's no problem. The tracking ID from the object tracker, the relative bounding box coordinates, and the file path that can be later used for post-processing. This slide, you can see some examples from our first field tests. So these are insects that are basically caught by the camera. You can see there are still some problems with shadows. So we are still working on this, how to deal with shadows on our flower platform. And most of the insects that we are detecting are wasps and bees at the moment. So we are also working on different flower shapes and colors, and also maybe scents in the future to attract more specifically, for example, hoverflies that we are interested in. Then, of course, the big question is, can we use this model output, so the crop detection, can we use it for classification in the next step? Because this is really important. We began with using iNaturalist to test if it is possible, and it looked good for most of the pictures. But now we are also using the new YOLO v5 v6.2 for classification. So we are also publishing a Colab notebook where you can train your own YOLO v5 classification model. And you can pick between different weights. So you can use the YOLO v5 weights, but also ResNet or EfficientNet weights as base or pre-trained model for your own classification model. And I think Kim Pierge and Toko Hoyle have some really interesting YOLO v5 classification models that maybe in the future could be also used in this workflow. So we can export this classification model in the ONNX format, which can be used for local inference on a CPU. So you don't need a special graphics card, and you can still run the inference on your laptop. In this example, I'm running it on my laptop, and it's fast enough to analyze basically hundreds of pictures in a few minutes or even seconds. And you can use the direct camera trap output, so there's no dataset preparation necessary. I modified the classification script a little bit, that it also takes the metadata CSV files as input, merges them, and writes the results from the classification in these metadata files that were generated by the camera trap and Raspberry Pi. So you will get as output the merged metadata CSV files together with the classification results. For example, the top 3 classes with the top 3 probabilities, but you can also adapt this to a use case. For example, you could use the top 5 classes, for example. And also in this case, the classification model is just like a module. So it's really easy to change the classification model with any other model. So at the moment, we are using a very basic model, but I think in the future, these models will get better and better for classification. Regarding the power consumption, in this graph, you can see the power consumption while detecting and tracking 5 insects and saving the crop detections every second for 5 minutes. The peak power consumption, so during this operation for the whole system, so the camera together with the Raspberry Pi is about 750 milliamperes on average, which is 3.75 watts, which is relatively low. So we can use the system during a whole season only by charging it with a solar panel. That's no problem at all. For the power management, we can set a wake-up alarm in the PyChoose CLI. So this comes together with the PyChoose board. For example, you can set several hours where the whole system will wake up for every day, for example, at 8, 10, 12, 14 o'clock. We can also use the output from the PyChoose board, for example, the charge level, to make the recording time dependent on the battery charge level. So if the battery is fully charged, we can record for a longer time, for example, 40 minutes. And if you have a lower charge level, the recording time in the steps that you can define will decrease. So we are using the system as efficient as possible. If you have only a low battery power left, we are using just a shorter recording time. This information gets written to a log file, so you can always check how the recording time and how the battery charge level changed over time. We can optionally also log a lot of battery information that we can read from the PyChoose board, and the temperature of the battery, the Raspberry Pi CPU, and the Oak CPU. This is not necessary, but maybe during the development phase, it's good to spot some errors or possible problems. Together with the Python scripts, we will also publish a documentation website where you can find very detailed information about all of the components that are necessary to build a setup. And you will also find detailed instructions on how to build the whole camera trap system, basically. But also information about the Flower platform that we are using. And I think this can still be optimized. So this is only like a starting point. You will find information about the setup of the Raspberry Pi and what you have to configure for everything to get to work. So it makes it possible also for, if you don't have any experience with Raspberry Pi or Python scripts, I would say it's still possible to use it together with this website. So yeah, thank you for listening, and I hope there's still a little bit time left for some questions or discussion.
Thank you much for an amazing talk. It gets better every time I hear it. So I am very happy to open the floor for questions. Again, you can type it in the chat or raise your virtual hand, and you can directly ask Max. There is already a short question, and they ask if the website is live now.
Not yet. I hope it will get live maybe this month or at least this year. So very soon.
Fantastic. I do have a curiosity, I guess. It's you used a plate with colored flowers on it as a landing platform and a trackpad, for instance. What was the material? What's the color that you used? And if you had different tests with other materials, and what's your feeling about those?
So the flower platform that we're using at the moment is just a normal foam cardboard. I think this is how it's called. So this can be used for example, for science, for outdoor use. But we are starting experimenting with different materials, different colors, to find the most attractive material and color for hoverflies. So hoverflies are our focus group at the moment. And I think, yeah, we need definitely a lot of experience to find the optimal material, the optimal colors. So what we're using at the moment is just like the starting point, basically.
Mm-hmm. Yeah, I'm really interested to see the different effects of the trackpads. So we have a question from Nildo Marini. What was the percent match at species level you got in iNaturalist compared to YOLO v5?
I mean, I just used iNaturalist to check if it's even possible to use the images for classification. I didn't do any more in-depth analysis, but I mean, it depends on your model. If you train a YOLO v5 model with a lot of data, I think it can get as good as iNaturalist, but you will need, of course, a lot of data, and iNaturalist has extreme high amount of training data. So I would say it just depends on your training data. Yeah. But Toko Hoy's working group and Kim Bjerge, I think they are releasing a relatively big insect classification and detection model also for the community. So this could be a good starting point for the future.
We have another question from Javier. How much is it each unit? How many have you deployed? And I apologize if you have provided this data, but I missed the 5 minutes of your talk. So I think maybe you have mentioned it. Perhaps you can remind everybody.
So depending on the components that you use, it's between 500 and 700 use. So with 500 use, you can already do everything. If you need more battery capacity and this mounting options, it will get to about 700 euros. And at the moment, we only have one prototype deployed because I'm using the second one for testing. So, but we want to start next year with deploying it on different sites, more units.
Yeah, I missed the question from Jen Young, which asked, is it possible to add a ruler and detect the insect size, which would help with the classification?
Yeah, and I think the next comment already has the answer for this question, because with the relative coordinates, you can just calculate the bounding box sizes if you know the frame size. So if the platform fills out your whole frame and you know your platform size, you just use the relative bounding box coordinates to very easily calculate the millimeter size of the bounding boxes. So this is really easy, it's no problem. But of course, it's only the bounding box. If your insect is smaller than bounding box, you only have an average size.
Would it be possible to detect the head position and tail position of the insect?
I think it's possible if you train the model for this. So if you train a model with data on insect heads and insect tails and use this model, you can use these two different classes to only detect heads and tails. But yeah, you will need the training data that fits this question.
Yeah, perhaps we'll take a last question and the rest can be answered by Max in the chat. So Quentin asks, do you use a single frame for each tracklet, or do you combine multiple shots to make a single average prediction?
I don't really know. I mean, I'm just using the object tracker that's in this Python API. So it's from the OpenVino toolkit. I think if it's possible, it uses every single frame, but if there's not enough frames for the tracking, it can track up to 5 frames in between the detections. If this is good enough of an answer, I don't really know more details about this object tracker. It's easy to use in 6 lines of code, but you can really go deeper into the configuration.
There is another question from David, and I encourage Max if you can answer that in the chat, but in the interest of time, perhaps we shall move on to the next speaker, which is Hjalte Mats Rosenstad-Mann from Aarhus University, and he recently began his postdoc there, if I'm correct. And I will leave the floor to you already, Hjalte.
Yeah, thank you so much, Luca. And thank you for the invitation. So I trust that you see my slides now, is that correct? And you also hear me?
Yes.
Yes.
Perfect. Very good. Okay, so yeah, you already gave a, a well enough introduction of me, so we can go over this very quickly. I recently finished my PhD at Aarhus University, and I worked mainly with developing methods for automatic monitoring of plant pollinator interactions using cameras and deep learning. And now I'm employed as a postdoc, and both of these endeavors have been with Toge Høje as my PI. I mostly work with plants and pollinators, but I would say that I'm in general interested in image-based monitoring of nature and anything that can, any technology that can aid or scale up ecological research. But this talk is about, firstly, automatic monitoring of pollinators. And I will start out by trying to convince you that automated monitoring of pollinators with cameras and deep learning is amazing. And I assume that I'm preaching to the choir here, so I think I can do this in just a couple of slides. And then we can move on till a small section I called but. I think there are some challenges with these methods, and it would be interesting to perhaps go over some of them and discuss them. And I want to illustrate one of these challenges with a case study from one of our projects. And after that, I will give some perhaps mostly inspiration on, I call it the dream, maybe it's just my dream, but let's see in the discussion afterwards, the dream of the universal insect detector. And then some acknowledgements, and we're done. So first of all, monitoring pollinators with cameras and deep learning is amazing. And Kim Bjerre has already been mentioned a couple of times with good reason in these talks. And here is some results where he was also the main protagonist. Here we built a camera system which included a small computer. And the computer was able to run a deep learning model. And this model was trained to identify 8 different insect species. And we also developed a tracking algorithm to track insects that moved within the camera frame. And you see some of the results to the right here. Here are track patterns and detections for 2 different species groups, bumblebees and honeybees. And you see results from 4 different months during the season. Hopefully, you can see that the patterns, they vary between these 2 groups. And you can probably also see that they vary throughout the season. And even though we didn't quantify this directly, I hope you can also see that these patterns seem to broadly relate to the resources that are available in the given time period. So you have some example images to the left there. So for me, at least, I don't need to see anymore. I can already conclude that this is amazing technology. It allows us information that would be entirely impossible without them and only relying on the traditional methods. They give us insights into pollinator dynamics that would be completely impossible to reach with other means. We also see that published deep learning models, they reach quite impressive accuracy for detecting and classifying insects. We've just seen from Max some examples of this. But here comes the challenges. Usually, these insect detection and classification models are trained on quite a limited number of species, and we can probably assume that the models do not generalize very well to other settings than they were trained upon. So there could be other species groups, other backgrounds, other natural settings, habitat settings, could be other camera systems, and so on. All these changes will usually create problems for the model. And I say probably because this is our experience. but it is something that is most often not actually tested in the literature because it is really difficult to test, and perhaps we don't even have data that we can use for testing this. So that means that if you are someone that has a lot of images and would like to go through them to search for insects, and you don't yet have a trained model for this, you have a challenge. The first step can be quite daunting and will usually involve quite a lot of manual work, especially if you have a lot of negatives, so a lot of images without insects in them, and you need to filter them out to actually find the images that contain insects so you can annotate them. So here's a case study, perhaps illustrating this problem, and also an example of how we solve this issue of manual labor in our case. So this project was about studying plant pollinator interactions in the Arctic, and here we use these time-lapse cameras you see in the top image. They point down towards the ground and they take an image of the vegetation below every minute or every 10 seconds throughout the growing seasons. We've deployed these cameras at a number of Arctic sites. You can see a time-lapse video from one of the cameras in the bottom here. Firstly, you see, hopefully there are a lot of flowers. They are flowers of Dryas specifically, and they were a main focus during my PhD. And hopefully you also saw some flickering in some of the flowers. And if you did, you saw some insects visiting these flowers. So in this project, we wanted to derive some information on flowers, flower phenology, and we also wanted to derive some information on the insects visiting these flowers. Now, for the flowers, we solved the challenge by firstly using a convolutional neural network to detect flowers in images, so we could count them. And we also developed a flower tracking algorithm, so we could follow each flower throughout a series of images, and we could derive information like you see here to the right. So here you have day of year on the x-axis and each individual flower track on the y-axis, and you can see when each flower bloomed and when it wilted and for how long it was actually a flower. You can see that there are some variation in when and the timing of flowering and for how long a flower is actually a flower. So that's great. We have some quite detailed information about the flowers here, but we also want to assign some insects to these flowers. We want to add some insect data to our flower data. And in our case, we did, as I mentioned before, have a lot of negatives, a lot of images without insects in them. Here are one of the very nice examples, because this is an image that actually has 3 insects visiting flowers. So this was a great day when we stumbled upon this image. So we were able to find some just looking through images ourselves, but we were not able to find enough so that we could trust that we could train and test a deep learning model that we would actually trust. So we needed some help to gather some insect data from these images. So here's what we did. We used our flower detection model on 7 different time-lapse image series. We detected all the flowers in all the images, and we cropped them out, and we created these flower collages, as you see on the right here. And we did this at quite a large scale. As I said, we did it on 7 images, and that actually produced more than 3.8 million flower detections or flower instances. As I said before, we also did have a small set of known insect visits to flowers, and we used these to create 19,000 flower crops that actually that we knew contained an insect, and we scattered those around our flower detections that we had not looked through ourselves, and created almost 1 million of the flower collages I just showed you before. And of course, we needed some help to look through these, so we created a citizen science project on a platform called Universe. I'm sure many of you are familiar with. The project is called Pollinator Watch. We uploaded all our flower collages and presented them to our users with the question, do you see a pollinator in this image, yes or no? And some results from this project. It ran for 211 days. And in that time, the volunteers, more than 4,000 of them, produced more than 3.7 million annotations. So annotations here are just answers to this question, do you see a pollinator or not? Yeah, and as you can see, quite a high number of volunteers. I actually checked a couple of days ago, and now we have more than, I think, 8,600 or something volunteers in this project. And it is, in fact, out of data right now. We cannot keep up with these people. They are productive. We had an average of 17,000 annotations per day, and our best day, in our best day, we produced more than 150,000 annotations. So, of course, the question remains, did we actually find some insects, or did these volunteers actually find some insects? And they did. They found more than 3,000 flower detections containing insects. And here we are only talking about what we call large visitors. So that's, as you can see in the examples here, syrphid flies and ladybirds, honeybees, beetles, and so on. And a detail here is that these 3,000 visitors, they come from quite a high number of crops that were actually flagged as containing visitors by the users. So what that means is that there were a very high proportion of false positives. In fact, I think they flagged about 100,000 collages as containing visitors. So there were quite a few cases where they saw a visitor in a flower, but where our expert review of their classification did not find that insect, or did not agree with that annotation. Now, you could maybe worry that maybe these volunteers are just really bad at finding insects in flowers. But as I said before, we also included some collages that we knew contained known visitors. And for these, the users were actually able to find more than 94% of them. So we can probably assume that the users are quite good at finding these insects, and that the 3,000 visitors here are probably, if not all, then at least close to all of the insects in the flowers in this data set of almost 4 million flower detections. So just to show you what could be done with data like this, I want to show you this plot. So it's very similar to before, day of year on the x-axis and individual flower tracks on the y-axis. Now you also see some yellow ribbons, and these just signify daytime, and they are there because these were the only periods where we actually looked for insects in this series. You also see an inlet, and that is just the proportion of flower detections for each day out of the total number of detections made for that series. And most importantly, you see some red dots, and those red dots are, of course, insect crops. So it's important to say here that each dot here is not a unique visit. So if you're looking at a cluster of red dots, it is most likely an insect that has been in flower for consecutive images in a row. So there are quite a few of those. We also have examples of visits that only lasted just one image, or only found in one image. I won't go into detail with this, but I'm sure you can see a few interesting patterns. I think one interesting thing is that most of these flowers actually get some activity from these insects. You can also see that there are some days with very low activity. There are 3 here, and 2 of them have we found no insects in. And this is not because of lack of data. We did have flower detections here, but we see this pattern in other series from the same site. So probably it stems from bad weather, which meant that there were no insect activity in those days. So we have a few of these series, but I will leave you with this small teaser here. And I will move on to my dream of a universal insect detector. Of course, when you work with millions of images and the insects are hiding in these images, it would be great if you and everyone else could download some general model that you could run your images on, and you could find the insects in the images. I will say that these next slides, they probably contain more questions than answers, but I was hoping that at least maybe they could inspire for some discussion later on. So let's see. First of all, how could we create such a model? One option would be that we all get together and we bring our data, our annotated insect data, all that we have, we pool it together, and we try to train one model or a few models on this data, and see if we are able to detect insects in just as insects, or perhaps just as very broad classes. So we don't want to classify, we just want to detect insects and then worry about the classification later. So this is quite similar to some other methods that are out there. I'm guessing that many of you probably know about the mega detector, which is very similar to what I just explained, but it's mostly on mammals. So it's quite a large model trained on a lot of data, and it's able to quite reliably detect the mammals in images. And it's used, people use them for like scanning through their wildlife cameras to see, to filter out the negatives and find the images that actually have something interesting in them. Now, of course, there are some questions here that we cannot answer before we have tried it. First of all, do we even have sufficient annotated insect data to do something like this? And I don't actually know the answers to these questions. I know we have some, and I know other people have some, but is it large enough data sets, and is it varied enough? Yeah, I don't know. That would we would have to test. And of course, even if we have enough data, does it even work? We don't know, but I think we should try. Now, one other thing we could do would also be to create our own data. We could build our model on fake data. So that would at least remove the question of whether we have sufficient data, because since we are creating it ourselves, we can just keep on going until we think we have enough. But it doesn't really answer the question of will it work? In fact, this question probably gets a bit stronger. I've just given you some examples of some fake insect data here. So the butterflies to the left are created by a model type called a GAN, a generative adversarial network. The insects to the right are created by a text-to-image model. So they are created based on a text prompt alone. And I did actually take just a couple of these insects to the right and scattered them around a very varied image set that is available online. And you can see this is not very realistic, of course, but it was done in an attempt to see if we could try to remove the challenges with the different backgrounds from these models. So the idea was that these images have very varied backgrounds. To be honest, I went into this with very low expectations, but to my surprise, this small model was actually able to find some real life insects. Now, this is just a very small test, so I will not say anything about to what degree this will happen. I haven't done any formal tests yet. We are scaling up this method now and also developing a better test for this. But as you can see, there are actually some examples here where the model successfully found the real insect in an image, even though it was only trained on completely fake insect data. You also see some examples here where the model failed, and one where there's something bad going on with the scale here. Okay, so I'm almost done, but I thought I would leave you with just a few bonus slides. And the point of these is that now I've just talked about a general insect detector, but I want to make clear that there are a lot of things we can do and a lot of things we can try and develop on our deep learning methods, even though they are only made for some specific site and some specific setting. I think there are a lot of improvements that we could make to the performance of these models. So one example here is just one slide here. It's work from Jamie Allison. I think he's also in the presentation here. At least he asked the question before. And this video shows a result of a background subtraction method. And as you can see, this filters out the information about the insect going through the frame quite well. And the idea here is that perhaps we should supplement our image data with something like this. So instead of just relying on RGB data when training our models, maybe we want to incorporate the information like this in our models. So I think that's something very interesting to explore. And lastly, there are also other methods I think are quite interesting. And one is that perhaps we should, at least in some applications, focus on learning the background instead of the insect. So this is just completely made up a result. I didn't manage to actually produce a result here, but the idea is that you could train a network to know the background, and when the network then sees something else, it's an anomaly to what it's used to, and that could mean there is something in the image which could be an insect. So this is something we are exploring now. I think, yeah, the result would be quite interesting. Lastly, I will leave you with these questions about the universal insect detector. Is it possible? And if it's possible, how do we actually do it? And if we do it, can we actually make it accessible? I think this is quite important, that even if we can make it and get it to work, it also needs to work for the wider community of researchers that would like to apply it. And maybe most importantly, do we want it or need it? And in my opinion, I think this is the most important question, because if we do want it or need it, I'm quite sure that we can, to some degree, make the other things work. Yeah. So a few acknowledgements, and that's all for me. Thank you for listening.
Thank you very much, Hjelte, for a wonderful talk, and I think very important questions as well. I would say that we can take 2 or 3 minutes of questions, and then we move on to the discussion session. So please don't be shy. I'm sure there is plenty of burning questions from the audience. You can also raise your virtual hand and and ask directly, Hjelte. While the people are starting to think about their questions and typing them in, I was wondering, with your citizen science project on Zooniverse, especially, you got the annotation for the whole set of 4 frames, correct?
Yeah, exactly.
So
they only answered the question, yes, we see a pollinator in that image, and it could be in any flower or several flowers or so on. And we took that approach to be able to have them process as many flowers as possible, and with the assumption that we knew that there were quite few insects, so it would probably be doable to go through these afterwards, and that's exactly what I did. I went through these 100,000 images very fast, and then checked where is the actual insect. Is it in the top right or top left or so on? You could also have just had the volunteers do it, but to be honest, we didn't really expect this much engagement from the volunteers, so we were really positively surprised there. They were, yeah, as you also saw, they really wanted to put in some work on this problem, so that was really great.
Yeah, thank you for that. I think that's a really powerful combination for engagement as well. Not too far from this note, Michael Bunsen asks, I'm curious from an organizational perspective, how can we all work together to create models, infrastructure, and devices? So these are complex questions, so Hjalte, give your 2 cents, and maybe we continue during the discussion.
Yeah, so I don't think I can answer that question, except that I can say that I think that we should get together, or at least if we do try to get together and formalize this work a bit, I think we will probably reach further, or at least reach there faster. It's not an easy task compiling data, having it in correct formats, and understanding what is in people's data. Training large models in itself is a challenge. So I don't think there are any easy answer to that. But I'm happy to see that someone asks that question, because I assume that means that someone would be willing to actually be part of this and contribute to such a thing. So that's great.
Michael adds that they're using insect data in GBIF, so I recommend everyone sending data to that service. And I also agree, consolidating data in one repository is a good idea. Tom asks, for the mini detector, the insect version of the mega detector, is there a lot of work to do before we can make that work? For example, standards, place to host data, etc. Or do you think we can get over those barriers quickly?
I think so. The famous last words for things like this is that we can probably do it quite quickly. We can do it in an afternoon, and usually it's not like that. But again, whether it will work to a satisfactory degree, I don't know, and I don't want to put my head on the line. I don't want to say that it will, but I'm sure that it will get us somewhere, and no matter what, it will give us some really interesting results, and maybe even more importantly, a fantastic data set to benchmark against when other people develop methods or train models. It would be great to have a compiled data set to use for pre-training perhaps, or to benchmark against. That being said, I think definitely we could overcome these the technical challenges. We can overcome if we are some people that have some data and want to do this. Surely these challenges are to be solved. No problem. Yeah.
Okay. There is a point from Malika that they on background subtraction.
Yeah,
I don't know if you want to comment on that, Hjalte.
Well, I know Malika's work, so yeah, I know about this, and it's a great work, great results, and very inspiring. So yeah, yeah, that's all I can say.
Okay, perfect. I think we will move on to our discussion session. So we are a little beyond schedule, but we will compress the time at the end to finish in time, hopefully. So now we will be separated in a number of breakout rooms. There will be a more experienced member of the Easy Rider, hopefully in each of them, so that they can help the discussion going. And afterwards, we will come back and present the insights gained from these group discussions to the audience. And we will finish at 5 p.m. So these are the discussion points. I will leave this slide on so that everyone can refer back to it, and feel free to pick one theme or multiple themes. I underlined my favorite question for all of this, but feel free to discuss any and all. And I think Alba can open the breakout rooms.
Created by the university. So the university set this up, sent a bunch of people from different domains to some beautiful cabin in the mountains and supplied them with beer, and had them discuss and try to set up some collaboration. And one collaboration that was a result of this was trying to extract insect data from weather radar data. And it was a perfect example of some radar specialists that were so annoyed about the noise in their data, and the noise are sometimes actually insects. And then an entomologist that really wanted to study insect movements at large scales, which these radars are perfect for. So I think, yeah, at least this example could maybe tell us that sometimes it's great if someone helps us start collaboration. We all agreed that these collaborations, that they work very well and are very fruitful when they are established, but perhaps it's good to get a push for actually establishing them. Or maybe we should also conclude that if you don't know what to do tomorrow at your coffee break, maybe you should go over in one of the other departments and ask around what they are doing over there, so we can get to know each other. Yeah, I can go on for hours, but I think we are out of time. Is that okay, Luca, or should I continue a bit?
I think I will quickly say some final words for the people that have parental duties or other important things to do in their lives. So we're not completely leaving everybody stranded, and then we can continue for the ones that can actually stay, which you're very welcome to. So when it manages to load, yep. So the last, well, thank you to everybody. And you can find the recording for this event on the same event page that that you find the Zoom link in. Don't hesitate to get in touch with us. So you now have the link to the slides. But most importantly, if you like discussing about these things, we have a group on Wide Labs, which is an amazing platform that is especially built for collaboration for technologists, ecologists, computer scientists, stakeholders, everybody. You can just get on there and get into the discussion and be notified for all of this. So you can just follow the link or scan the QR code. We're not very many so far, but more content is coming, and we're definitely there to answer your questions. So with that, I will stop sharing, and I thank everybody that has stayed so far, and we can carry on with all the other summaries, and then yeah. So who was number 3?
Max,
I
yeah.
mean, we didn't talk so much about data standards, but Jamie shared a really interesting link, and I posted it also here in the chat, ChemTrap DP. Maybe Jamie, can you say some more details about this?
Yeah, so I went to this webinar yesterday, a GBIF hosted webinar about camera trap data sharing. And there was quite a lot of talks related to this standard system. It's called ChemTrap DP, and it's kind of it's like Owen Core type metadata system. So it's a way of describing your data from your camera trap, which will start with your project, And then underneath your project, you'll have your individual camera deployments. Within each camera deployment, you'll have your image series, and in each image, you could have potentially several organisms. Basically, it seemed like it was pretty developed and potentially, yeah, a good starting point. If there's anyone out there, maybe a bit like me, who's thinking, how should I be? If I were to document things exactly the way I should, what would I do? Yeah, and it's the point where actually the other GBIF guys who are developing the universal standards are now trying to, it seems, adjust to this CamTrap EP system, because there's so much uptake of the in the camera trapping community. So I want to bring that to people's attention, because it looks like it might have some future to it.
Yeah, definitely to keep an eye on. Rule number 4 was
Hi, Luca. I think that was me.
Yep.
Is that right? Yeah. So, hi everyone. I didn't realize that the notes I was making during our chat were going to appear in the chat. So, we talked about the questions were around how to attract different insect groups, dealing with taxonomical bias, limitations in methodologies, and how to standardize insect monitoring across the ecosystems. Quentin talked about his experience with using AI from sticky traps in the field, and being able to combine different predictions from different frames of the same insect when it was constantly moving positions, and also the challenge of hierarchical classification problems, so that yeah, the sort of depth of taxonomy wasn't sufficient. And that related to again, us talking about a little bit about species complexes and the very yeah, challenging nature of some basically what is a species. And in that situation, maybe DNA monitoring is a more surefire way of understanding operational taxonomic units, or a measure of genetic diversity would at least give you something a little bit more concrete in that sense. But an interesting theme that we finished on was around the kind of challenge of standardizing. And yeah, I was really interested to know from the guys with more experience here, whether anybody's actually looking for a more formalized index that's a kind of equivalent to the genetic operational taxonomic unit that you'd get from DNA barcoding. Again, Quentin was talking about concepts maybe where we could start to think about generating, you know, regardless of understanding what species we're seeing on these camera traps, indices of morphospecies richness or size, you know, some kind of composite index of size, color, behavioral richness as a standardized measure. If it's the case that across countries and continents, we can't come up with, you know, the same taxonomic resolution that would be to species level, which would be the absolute gold standard. That was, yeah, that was probably I think probably the most interesting.
Yeah, that's a tough one. But I think we definitely should be working towards something more all-encompassing, some universal, to have at least a conversation across ecosystems and across taxonomic boundaries. But how that looks like, I cannot quite see at this moment. Perhaps somebody else has some insight.
Should I go with group 5?
Yeah. Room 5, Tom.
Yeah. So we decided that we want these things in the kind of idiot-proof format that ecologists can use, kind of plug and play in the field, and we'd like them now, please. We talked a little bit about the value of models on the system, the edge processing, and in the lab, and what do we need where. What do we really need to work in the field, and what can kind of hold off until we've got things back in the lab. And we talked a little bit about the big data problem that these systems can produce, depending on their settings, can produce very large amounts of data. And we saw the value in running these models on the edge. We saw a lot of value in helping to kind of reduce that big data problem. We also talked a bit about, in terms of the very first question on your list was about the hardware requirements. And we kind of were highlighting that that's actually really geographically dependent. And so Joe, who's working at high latitudes in Canada, has problems with sunlight, which other people close to the equator don't have. And it also kind of follows on to, you know, people in some parts of the world will find it difficult to get access to GPU units, and even turning to Google Colab wouldn't be enough to satisfy, you know, larger projects. So I think that's kind of an important thing to think about for those who are, I guess, developing models and wanting people to use them. So thinking about those communities where people don't have access to these sorts of processing, how are you going to make your model available to those? And for people who are developing and designing hardware systems, just to think about locations outside of Europe and North America, and how the challenges that people have in other locations and how those designs will be able to respond to those needs.
Yeah, I think very important points. So the last room that we have was Brian, Kim, Mark, Belial, and Roy.
We didn't stick particularly well to the agenda, because when we went into the room, we realised that we couldn't see the themes anymore. But a lot of what we said has already been covered. We were talking quite a bit about this problem of agreeing on data storage. Brian mentioned he has a big project to identify bumblebees, where he's had thousands of images, and that's now been sort of rolled up into an app. But again, there's this issue of making sure you've got good annotated data sets to work from, that you need to train up the models in the first place. And then if you do get people, if you get citizen science people to do the annotation for you, you want to make sure that those data are retained and available for other ecologists to use to train their own models from, and build on the work that you've already done, so that it's not lost for the future. So I think those were the main things, unless anyone else in the group can remember anything else.
Yeah, I think we covered a lot of ground, like really a lot of ground with these discussions. It was intentionally broad to let everybody pick and choose what they preferred. What I'm especially curious, well, as a tool developer, I suppose, it's where can we make the most impact with these methods? Like, how can we reach more ecologists? Because I think that success stories of collaboration, interdisciplinary collaborations, has been to reach out to other people and get them engaged in these ecological problems. So rather than just publishing in a computer science journal our mediocre model that works more or less how we desire it to be, but we can publish it in a much more like field-related journal and maybe get more use out of it. Like, people can get more use out of it. So I'm wondering, like, I would like to pick your brain on where would you look for these things? Like, where would you look for these methods? Do you scour GitHub, or you look for publications, or you Google it up?
Yeah, I guess not quite answering that question, Luca, but I also think it's about not just making the model code available in the paper. It's about like hosting that model somewhere where people can really dump their images into it, kind of like Megadetector do something a bit like that, right? So then you don't have to have the R or Python or TensorFlow knowledge to use it. It's just like, you know, I'm a wildlife manager. I manage this small reserve, and I can just now take these images that I've got, and I can chuck them in. Yeah.
Yeah, so how do other people feel? That's they're willing to put in the effort of learning a framework and deploying their own model, or a web-based app where you can drop your images and not do much else would be enough?
I think there is something maybe in the middle, which is
Yeah.
having APIs where you have a web platform that you can send data programmatically to, and it sends you classifications back. So it's kind of, you don't need to know everything, but you can still do it automatically.
Mm-hmm.
Yeah, I think something in the middle is the right idea too, because I think the web interface for classification maybe seems practical, but the initial data filtering maybe, I don't know, for example, if you have terabytes of images, raw images from the cameras, and there's been no object detection or data filtering, are we really going to have a service where everyone is sending all of those raw images, you know, on potentially across the world to, you know, to one data center in Canada, so I think something where we have some data filtering and perhaps object detection on the device or offline, and then a centralized service for the classification might be one place to break the services.
So we just have to convince iNaturalist to give us their API to just let us use their model. No, I'm just joking, but I think it's a good point. Yeah.
There also is, with the insects, we're finding, it is different than with the mammal images, because you have maybe you could have between a few to hundreds of insects in a single image, and with the mammal cam traps, I know some people here that collect the images, and they get maybe they go up there every week, and there's maybe a few hundred images in their cameras. But here it's like we're getting 7,000 images in one night, and each image has multiple insects in it, and they're all very small in the image. So it does have its own challenges, the insect monitoring, and the iNaturalist I don't think is even set up for. One note on that though, the iNaturalist is currently taking advantage of the free image hosting from Amazon if your images are under a public license. And some people are having trouble using this service because they didn't track the licensing from the beginning of their images. So they don't know whether they can put them under an open license. So if you want to think about that now as you're collecting images, if you are okay with putting them under an open license, then we can at least start, even if they are terabytes of images, we could stuff them in Amazon's S3 service. I'll note on that though, you still have to pay to process them, to get them out. The bandwidth is not free. So be wary if you put 100 terabytes in there, if you want to process them, even run them through a classifier, you either need to pay for service in Amazon, so you can use the images within their network, or you have to transfer it to your own network, which it does cost money. I do like this RoboFlow service. I haven't tried yet, but one speaker showed today for annotating online and crowdsourcing annotations.
Yeah. Yeah, I haven't thought about the licensing of images. That's actually quite a good point. We should probably be thinking about that before putting the cameras out, and maybe coming up with a solution before it becomes a problem. Yeah, also Tom's point about ecologist probably is a bit of too broad of a category to actually cater something to their needs. So maybe coming up with different user profiles, like a researcher versus a student versus a stakeholder, is probably the way to go for try to make something useful for each one of these audiences. And I'm not saying that we should make it for all of them, but at least we can like divide and conquer, so to speak.
But yeah, I think I kind of agree with Quentin. Like the API route is kind of agnostic in a way, because then you can build an R package that wraps that. Developers can use that, and then you can leave it to developers to build the GUI or whatever that works with it. So I think it's good. Yeah, good way to go. I'm gonna have to head off, Luca.
Yeah, I was about to say.
And I think it sounds like the sort of conversation wrapped up. So I just wanted to say a big thank you to you for putting this all together, and Alba. It's been really great. We had there are loads of people on the call. I think we've had some really good conversations in the breakout groups, and all the talks were fantastic. So thank you for all your efforts on this. And you said the recording will be available and it'll be on Wild Labs within a week or so. So if you want to share it with others, and send them there.
Yeah, thank you all for joining. Alba, if you have anything to add, now's your chance. But
no, I was just wanting to say about the recorded, but Tom just said so. Yeah.
Okay, perfect. So I really strongly encourage anybody that is even remotely interested in this to come and join the Wild Labs discussion. We promise not to spam you too much. And you can continue all of this and have like a more broad, maybe articulated discussion in discussion posts, so you can get access to all of the resources. I think it's a really great platform to converge towards. So thanks to everyone, and sorry we went a bit over time. Hopefully it was worth it, and we'll see you at the next workshop.
Thank you very much. Cheers.
Thanks,
Thanks,
guys.
Luca and Albert.
Cheers.
Bye-bye.